In the previous talk, have you heard about the analysis of prokaryotes? We summarize the differences between the eukaryotic genome and the prokaryotic genome. First, eukaryotic genome have exons and introns. Introns are removed before translation, so we have to consider this RNA processing when we annotate gene structure. Second, the eukaryotic genome has a large number of copies of similar genes and repeat sequences. Last, in the case of diploid or polyploid species like cats, they have haplotypes. It means that different alleles or different sequences exist on homologous chromosomes. Thus, the eukaryotic genome has a more complex structure than the prokaryotic genome. Generally, the assembly and the annotation of the eukaryotic genome are complicated and they require much time, money, and human resources than the prokaryotic genome. Now, we will start to talk about how to analyze our domestic cat genome as an example. Here is an overview of our analysis. This genome is a female American short-hair breed cat. Her picture is at the top right, with a size of 2.5 gigabases as a whole genome sequence. The number of scaffolds is 20, and we are able to reconstruct chromosome, in other words, pseudomolecule or chromosomal level genome sequence. We summarize the methods at the bottom of this slide. We obtained a large number of long reads by PacBio and Illumina short reads to correct the long read error, and Hi-C and optical mapping by Bionano Sapphire linked the assembled contigs for chromosomal level reconstruction. This project is facilitated by an international collaboration between Anicom Advanced Medical Research Institute, Katsa DNA Research Institute, the Chinese University of Hong Kong, and the National Institute of Genetics. Our team is responsible for gene annotation and database publication in this project.